Fabulous. Hi, everyone. Thanks for coming. So this is our 101, how to get started with brain imaging at Sinai. I'm just going to run through, since we're recording this, I'm just going to run through everything and then take your questions afterwards. But our vision for these kind of meetings in the future is that they be maximally useful for you guys. So I think in the future the format will be a lot more informal and it'll be much more of a back and forth question and answer thing. So how to get started with brain imaging at Sinai? Well, the first thing that you need to know, in fact everything that you need to know is on this website, https://bic.mssm.edu. So you'll be able to find everything else, so that's the only thing that you need to write down today. So what do we mean by brain imaging with BIC at Mount Sinai? Well, I'm mostly going to be talking about MRI today. So that's, it would be at 3 Tesla, 7 Tesla, or small animal imaging. But in addition, we could also class into that other kinds of imaging like positron emission tomography, PET, PET imaging, or EEG, which is something that's currently in development. And if there's anything else that you want to use and you don't see mentioned here today, then you should talk to one of us about whether or not we can use that, because we're always looking for new developments. So very basically what we have available from the brain imaging, which is well established at the moment, is a 3 Tesla Skyra MRI scanner, a 7 Tesla MRI scanner which has a human-sized bore, a PET MR scanner, which is a very cool piece of technology that combines the two, and some of the individual labs are using EEG, particularly the NARC lab right now. So I'm just going to talk you through how to get started with any kind of imaging projects that you want to get set up at all. And so the main thing that you want to do is visit this link here. Yes, amazing. Let's see if we can make this a little larger. So this is the Brain Imaging Centre's website, and everything that I'm going to say to you today is contained on the website. So here's the main link for starting a research project. And the main thing that you need to know is that there are 2 levels to this website. So there's a level that everybody can see, and then there's a level that's available to those of us who are at Mount Sinai. And in order to get to that second level, you need to create a login. And I can't see the login here. But here's the link to request the account. So it's right at the top of the Starting a Research Project page. So if you click here, then you get taken straight to a contact page where you can create an identity for yourself. So you just fill out the form with your name, email, a message saying please give me access to the website, and then you'll be able to create your own login. And for now at least, this is not part of the single sign-on, so you'll have to create an independent password that's different from your single sign-on password. So that's the only thing that's slightly clunky about it right now. So once you've set that up, then you'll be able to access all of the areas of this, including all of this information about how to start setting up the research project. So the basics are here for you. You do need an approved GCO number. I doubt there are many people in this room that don't know how to do that, because we've all had to do that for studies of every sort. And if you have human subjects involved, you will need an approved IRB protocol. If you have animal subjects involved, you need an approved Icahn protocol. In addition, if you're using human subjects, you need to create an MRM number, which you can do on the Mount Sinai intranet here. I'm not going to do this today since I don't have an approved IRB protocol. And you'll need that number in order to fill out the form and get started with your imaging project as well. So this is a number that you'll use to enter your subjects. I'm going to come back to this scheduling link later. First things first, you're going to want to fill out the survey. And I thought maybe the simplest thing to do is to visit the survey page together. Now this is a long, clunky link. There's also a link directly from the BIC website from that Getting Started page that I showed you, so it's all very straightforward. When you visit there, you're going to see a very nice BIC project startup form, which starts by saying thank you for engaging with BIC for your new project, and asks you a few questions. It's really, really important that you fill out this form before you start an imaging project. This basically supersedes any kind of forms that you used to fill out to do an imaging project, like the form that you would fill out for TIMI. So TIMI are part of this process, and any relevant information will be forwarded to them via this form. So this is now the form that you need to fill out. So first of all, it asks for your GCO number and the approval date and expiration date of your GCO. Then it asks for your name, principal investigator. So let's just fill out from here. So that's me. I'll enter my email address. Please provide the name of the study coordinator. Okay, so that's Bob Robinson. And the email address for Bob Robinson, I just made him up. Please give a brief descriptive title to refer to your project. So we'll call it MRI Study of Neuroscientists Viewing Brains. I've always wanted to know where the brain recognition area in the brain is, so that's what my study is going to be on. And a brief description. So neuroscientists will view different brains and receive functional MRI scans, something like that. Besides the PI and the study coordinator, please list other key personnel involved with this project. So that could be anybody in your lab, or it could be somebody from BIC that you've spoken to who's going to be a key part of your study design. And I'll come back to those people at the end and who those key personnel might be that you would want to talk to. Then it's going to give you a bunch of choices. The first one's easy. What category? Is it human non-patient, human outpatient or inpatient, vulnerable population of humans, non-human primates, non-primates, so other animals, or a pilot project. So I'm going to check pilot project in this case, since I don't have an R01 for this. And then it's going to ask me for my potential funding sources. If I were to just click human non-patient, then that box would disappear, I think. If I uncheck this guy here. Yes. Okay, and then it's going to ask a bunch of questions that you may or may not know the exact answers to, but it's important that you put some answers so that you can have a discussion about it. So what's the expected number of subjects that will have brain imaging scans? So you may or may not know how to calculate the power of your study. So if you don't know, just put something that's in the ballpark from someone else's publication. So I'm going to scan 40 neuroscientists. How many brain scanning sessions will be scheduled per subject? So in my case, just one. How many minutes will each subject scanning session last? Again, you may have no idea about this, so just put something in the box for now. So I'm going to put 35 minutes. How many days per week or month need to be scheduled for this project? Well, neuroscientists are pretty busy, so I'm going to say 1 day per week. And if there's any scheduling constraints, here's the time to do it. So for example, if my neuroscientists need to view brains 2 days apart or something like that, that's where I put it in. How long a calendar period is this project anticipated to acquire data for? So it lists numbers of months up to a year. So let's say it's going to take me about 6 months to acquire my data. And then it's going to ask you a little bit about the brain imaging facilities. So here's where you need a little bit more background. And again, you can go right back to that BIC page in order to find out what's available to you. So there are some supporting resources available in addition to the scanners themselves. One of them is the mock scanner here, and I've placed a link at the bottom so that you can visit later and find out more about what's available here. So essentially, if you have a patient group or a subject group that might need some training to actually perform in the MRI scanner, but you don't want to pay for full amounts of MRI scanning time, this might be a really useful resource because it looks and sounds like an MRI scanner, particularly useful for studies involving kids. In addition, there's a neuro prep room where you can train your subjects on their tasks, and you can also acquire some physiological data. And then there are a bunch of supporting resources that are available to you both in and out of the scanner. So I'm not going to go into tons of detail about them now, but we can discuss them a little bit after I've finished this. Basically, there's response buttons so that they can respond to a task, skin conductance, respiration, heart rate, pulse oximetry, eye tracking, EEG I mentioned already, anesthesia, E-prime tasks, and we have some other programs available too. Video and auditory stimuli, so there are a lot of different things that you can use in your study. And again, if you're not sure, just listen for now, and you'll be able to have a conversation with somebody about it. In addition, you need to know what kinds of scan are available. Now, theoretically, any kind of scan that you want is available to you, but here at BIC we have a standard protocol that we've spent a lot of time developing, which we highly recommend that you use for your data acquisition if you can. And you can use any or all of these things. There's a couple of reasons why we really highly recommend that you use these protocols. Firstly, because they're really well optimized, and they're very, very similar to the protocols used with other really high-level projects like the Human Connectome project. So they're going to give you great data. Secondly, we've developed some pipelines to process those. So right now, some of your data will be pre-processed and delivered to you in a better, more processed format with basically no extra effort from you, which is also great. And thirdly, many, many other people will be using them. That means there's a community of people to call on when you want to know how to do something. It also means that you could data share in the future if you wanted to, which would also be super useful. So I think there's a lot of reasons to use these BIC standard protocols. These include T1 and T2 weighted images. So these are your structural images, which are sensitive to different tissue types. They're good for all kinds of gross brain anatomy, and they're also used to align the other types of scans to each other. We have bold fMRI scans, so you can use this when your subjects are doing a task or when they're in the resting state. And these are really, really high spatial and high temporal resolution compared with other bold protocols that are out there. And they give you data on the whole brain. So again, if you can do this, I recommend that you do, because the whole brain is better than part of the brain. And we have a diffusion scan that's available, which will give you measures of structural connectivity and white matter integrity. And this will allow you to use most of the major diffusion MRI or DTI protocols that are out there to analyze the data. A few things that you need to know. So the entire BIC protocol, if you chose to acquire all of the data, not counting your task length, which might vary, would take about 35 minutes. So you get a lot in quite a short amount of time. It's available for 2 coils, a 32-channel head coil and a 16-channel head coil. There are a couple of differences between these, such as the size of the head that you can fit inside of the coil and the quality of the data that you get. So it's always worth having a discussion with someone at BIC about which of the coils is best for you to use. But the BIC protocols can be used for either. They're optimized to be HCP compatible. So I mentioned the Human Connectome project, which is one of the biggest projects looking across different subjects and using a data sharing program. So our data are very, very similar to these. They're very high resolution and they give us really great functional brain mapping in the bold sequence that we have. So that's definitely a plus. And there are a couple of things that you need to include if you're going to use the BIC protocol, which you will be reminded of, including a localizer and field maps that you'll need if you're going to do the functional MRI bold protocol. So just a few things to be aware of there. And here's a quick summary, which I'm going to skim over for now, but you'll be able to access from the website in the future, just showing you what the differences between the 16-channel and the 32-channel versions of the protocol are. The take-home is it takes about 35 minutes to get all of that data. In addition, you're going to be asked in the next box what kinds of analysis you might want to do, and so it might help to know a little bit about what's out there. So with your structural data, your T1s and your T2s, you might want to quantify the volumes of tissue types or the shapes and sizes of structures and how they change. So you can do things like separate out grey matter from white matter from CSF. You can look at the structure and shape of subcortical areas or cortical regions, the thickness of cortical areas, and you'll also be able to look at local grey matter changes, for example, between groups or over time in a longitudinal manner. With your bold fMRI, there are two main types of analysis available: task-related one, where you need to input your task parameters, and resting state functional MRI and connectivity, where your subjects aren't doing anything at all, but you might be interested in the intrinsic correlations between those regions, and again, how those differ between two groups. And for diffusion MRI, we can do things like tensor fittings so that we can get an idea of the underlying white matter structure, measures of things like fractional anisotropy that tell you things about the integrity of the white matter, and things like tractography, which will allow you to do things like segment different grey matter regions out and so on. I'm skimming over this really, really quickly, so anything you want to know more about, please ask questions at the end. What if you want something non-standard? Well, that's available too, and these things are in different forms at the moment, and they'll proceed with different speeds depending on what people are interested in doing. So if you're really interested in doing something new that I haven't mentioned in detail, please let us know. Some of the examples of things that you might want to use might include spectroscopy to look at things like GABA and glutamate, arterial spin labeling, pharmacological fMRI, real-time fMRI, high field strength fMRI. I mentioned the 7 Tesla scanner already, and it's a great resource. Multi-echo fMRI and non-human primate imaging, to name but a few. And I mentioned small animal imaging earlier. That's another one that should definitely be on this list. Okay, so once you've filled out the form, then you just click submit, and it will be submitted to the BIC core personnel, including Rita Goldstein, who will take a look at it for you and get in touch to find out what your needs are, and to invite you to come and talk to us about what you want to do in order to make sure that you've really optimized your parameters and set up the study the way that you want to set it up. Once you've done that, then you're ready to go. All you have to do is create your project. So in this case, we use this app called Calpendo. So you'll need to visit the link here and create a project in Calpendo, which requires you to just replicate some of the details that you put into the previous form. So it'll ask you for the PI, the contact details of the scheduler, the project title and description, how many hours you want, and so on and so forth. So I won't repeat that. And then, of course, you need to do your MRI safety training. So whoever's going to be running the scans from your lab needs to make sure they've attended MRI safety training so that they're okay to be around with their participants. Now, you won't need to run the scans yourself, because when you book the MRI scanner time, or when your coordinator does, then there'll be trained radiologists available to run the scan for you, and that will be something that's available to everybody who's scanning here. So you don't need any specialist training. This is just basic safety training. So it's just a short slideshow telling you about the dangers of MRI, and then a test just to make sure that everyone's comfortable with that. Okay, now we're running into a Windows Mac incompatibility issue. What happens to my data? Apparently it goes into a black hole. So what this did say is, once you've acquired some imaging data on one of the MRI scanners, then it gets pushed into a centralized server called XNAT, where your data is stored and backed up, so they're safely kept. You'll be able to access them in their raw format, and in addition you'll be able to run some pre-processing pipelines to get your data out in a usable format that includes some corrections for some of the artifacts that you might not want part of in your data. And I think the detail of that is really the subject of another talk, so I'm not going to talk in a lot of detail. In addition, those data will then get pushed onto a central server. So right now they're getting pushed to Minerva, and I think that's probably the way that's going to proceed. So Minerva, in addition to being a supercomputer and allowing you to manipulate your data, will also allow you to access those pre-processed data and take them off and do whatever you like with them. And over time, we'll develop this more, so there may be more things available to you, like even some automated analyses might get run on your data in the future. At every point, your data are your own. You can take them off at any time and work with them. You can access them in any format that you like, and ultimately, it's up to you how you want to analyze your data, publish the results, and get huge amounts of funding. But we're trying to support as much of that as possible. And again, if you use the BIC standard protocols to acquire your data, we can do that more easily. Oh, here it is. Look at that. Who knew? Okay, so it goes from the 3T scanner to XNAT to Minerva and the Timmy and Bic specialised servers, which at the moment are both doing similar jobs to each other. And then finally, at any point, the data comes off to you, the investigators. Okay, so if you want to know anything else, I really, really strongly urge you to come talk to us. These are the BIC core, so these people all have a great deal of knowledge about a great deal of things, and so you should really feel free to come and ask them about anything that you need to. And in addition, we have core administrative personnel who will help you with all kinds of issues, from scheduling to finding out who to contact about what. So you should feel free to contact them as well. In addition, if you want to know more, we have a weekly meeting which happens on Mondays from 12 till 1 p.m, just directly upstairs from here. It's a great place to bring your new projects if you're thinking of trying something new and you want a bit of advice and you're not quite sure who to contact. It's also a great place to stay in touch with the community and make sure that you have some contact with us. So I strongly urge you guys to drop into those meetings whenever you feel like it. There's no commitment to come to all of them, just come whenever you need to, basically. So that's all I'm going to say for the presentation part of this. So thanks very much for listening, and I'd really love to hear your questions.
Can you tell us about small rodent imaging? What's ready here?
Yeah, Lazar, do you want to take that question?
So there is a 7 Tesla and 9.4 Tesla available MRI scanners. And there is also ultrasound system, and there is a fluorescence system available. And now to be more specific about the question. The scheduling is basically very similar. So in terms of the creation project, it's all very similar to humans. What was described. The same procedure is very similar. Just it's location is different in different form.
Yeah, really interesting. Let's say the animal model for neurodegenerative disease, let's say Parkinson's disease or Alzheimer's disease. The imaging can change. Let's say the dopamine using the small the micro pad. I don't know if this.
Yeah, that was basically the there is no PET available, so there is a push to acquire it, but somehow it didn't happen.
Okay.
So there is no PET, micro PET. Yeah.
But for studying degenerative diseases, structural MRI is available. We use diffusion MRI to
spectroscopy. So there are alternative techniques, and if you're interested, we can try to discuss this.
Yeah, I mean, we definitely haven't forgotten about micropet as well, and I know there is a group that was interested in acquiring one that's part of Timmy, and I don't know where they got to with that. They're still working on it.
Instruments are pretty capable. They're very well used. They're well maintained. So, I think, are there any current Parkinson's projects running on CMT?
No. Yeah, so you could use functional MRI for some of this, for sure. So yeah, definitely talk to Lazar more about getting that set up. Thanks.
But we don't have a cyclotron. There is no cyclotron here, so we cannot do any ligands. If you're interested in dopamine receptors or transporters, anything with C11, carbon 11 type, we cannot do here.
So any neighbor institute that you know of now?
Yeah, neighboring institutes and through collaborations, yeah. But here we do have the PET-MR, but it's not for small rodents. It's not micro-PET. But it can be used for humans or medium-sized animals. But again, only F18 stable ligands, not C11.
Yeah, so you might definitely want to look into using fMRI and structural MRI. If you have a cohort of animals and you can scan them, then that would be pretty awesome, I think. Both of those scanners have really great resolution, so it would be great for rodent studies. Yeah.
Do you have any, the BIC standardized sequences is for adults. There's some functional testing in it. Is it for adults?
I guess it's designed for adults, but it can be used for
Modified
kids as well. Yeah, and I think there are groups scanning kids. Yeah, I think so. I don't think there's a great deal of difference between the protocols for adults and kids. So they could easily be adapted for kids. In terms of functional tasks, so the sequence itself can be used with any functional task that you have in mind. What people have been doing mostly to date is designing their own tasks and using the sequence to acquire the data. And so I think we have various stimulus presentation programs that we can use depending on what your lab is used to working with. So I think we have presentation, ePrime, we have MATLAB and Python as well, do we? I think. Okay, maybe not Python. I don't know anything about Python. Okay, okay, and yeah, we can install other things as well. I know.
Development.
Yeah.
The real-time motion kind of correction. Dr. Howland is working on that. It's not yet set, but it's something that can be really useful for imaging. Yeah. And also, depending on the PI and how many studies we have for kids, we can change a little bit the room to be more kids-friendly, and then remove it from the room. But that's what's being done in centers where we have a huge bulk of kids in the room.
Yeah. The mock scan is also super useful for kids because you can get them in and give them a practice run first, which makes it a little bit less daunting for them to go in the main MRI scanner. But yeah, I think you should be able to adapt those sequences almost straight away into kids, which would be really awesome. More questions? Yeah.
So if we want to do go, no-go task, which lasts 19 minutes, so the total time will be 1 hour, 19 minutes plus 35 minutes in a scanner. Is that how that would work?
Well, it's really up to you. So you can choose to incorporate different aspects of the BIC protocol depending what you need. So if you think that that's too long for your subjects, you could miss something out. For example, if you have a go, no-go task, maybe you don't need resting state fMRI as well. And so you could choose, how long is a resting state fMRI sequence? 10 minutes. Yeah, so you could save 10 minutes by just doing task-related fMRI. And you still get your diffusion imaging and your high-resolution structural scans as part of that. Yes.
And we have set recommendation, we can send that actually, where we recommend either resting state or DTI diffusion to keep it under 1 hour if you have a task. So you can choose either resting state or DTI, and also you don't have to do the T2 image. So those are the standard recommendations we give. And then that can fit within the hour.
Yeah. And if you were really struggling for time, you maybe don't need DTI as well, but of course, yeah.
And who is, I guess, is a data gatekeeper?
I guess that would be Chen, who's the data gatekeeper.
That's only temporary.
But it is temporary, yeah.
Setting up a portal where you'll be able to submit so the data are collected on XNAT, and from XNAT you have the option to download the data directly, so you can have it immediately without accessing a gatekeeper. And then there is no gatekeeper. You can basically get it from XNAT directly. There's nothing stopping you from getting your own data. And then,
in DICOM form.
In DICOM form, in the raw form. However, if you want, we also have facilities to process the data further, so that you can also access from XNAT. There's basically, Chen has set up a reference for how to, for example, enable to start the functional preprocessing or the anatomical preprocessing. And the last bit that we're constructing right now, which should be available soon, is a web-based interface to get the preprocessed data. So the raw data you can get right now, and then there's already a system to initiate preprocessing of the data, to remove artifacts, and to normalize spatially to get all the brains into a standard space. And then the last bit that we're building right now is the downloading mechanism of the preprocessed data. So we're going for a no gatekeeper model.
And then let's say, so that's our own data. And if somebody else is doing also effective no-go task, then it's investigators talk to each other and it's a collaborative basis. Is that how it's perceived or conceived?
The vision is, yes, is to put everything together. That's why we are recommending using the standardized type of procedures and acquisitions and adopting everything that's standardized. In terms of the tasks, people usually, PIs have their own tasks, unless they use HCP type tasks, the Human Connectome Project, and that's something we can help with.
Right.
And if there are enough PIs using the same task, we can definitely put that together, there is agreement, and then there can be data sharing.
So we sort of envisaged a two-tier approach to this. Initially, we'll just talk to each other, and if we want to collaborate with someone and use the same task, we'll do that. And I think going forward, the plan would be for us to have an agreement that we choose to sign, to say, yeah, I'm in. I want to share my data with everyone at Mount Sinai.
That's not in place yet.
But that's not in place yet. Yeah, so I mean, if you're keen to do that, then that's awesome. And definitely the first step would be to use the BIC standard protocols, which is what we're going to be using going forward. And to let all the other PIs know. We have a pretty good mailing list distribution system where we could let everyone else know, I'm thinking of doing a Go task. If anybody else wants to use it for their subjects, then that would be great. And we can certainly get that rolling really quickly. And then in the near future, people will be able to opt into a much bigger data sharing system.
We have some tasks already available, right? Lazar, I think we have some adopted ones. Which ones are they?
It's basically go no go, but it's called differently. Memory task here, I think it's called. There is a motion task. There is some. There is a language task, but I don't know what that actually entails. To tell you the truth. And there is one more.
Okay, maybe that would be something to add to the website, the details of those tasks.
Yes.
It's definitely something to
So,
Yeah.
so there is a version which is supported by BIC, and there are some people, if they don't like what is supported, they make modifications, and basically, there is one which is supported.
Just to add something. For the past, for the past year, BIC has focused firstly on the REST state data and diffusion and structural, because those are the aspects of data that are basically acquired in a similar way. They're relatively easy to acquire and process. Going forward, and depending on the interest levels, BIC is more than happy to assemble tasks, standardized tasks for the community to use. But to really do that effectively, we need as much input and feedback as possible, such that if we can, for example, if down the line, if there's a common interest in studying emotional faces or go, we can get together in one of these workshops, determine, so this is what Paula was saying before in terms of opening a dialogue and having a discussion, we can decide on the parameters and implement the task, either modify from an HCP task or take it from somewhere else. But this is the kind of interaction that we're going after. So that's more than possible. We just need to know what the interest levels are and what models people want to follow. We can implement them.
Yeah, that's cool. I definitely would be interested in that sort of thing. So I'm interested in memory, for example, so I would be interested in sharing whatever tasks I come up with anyone else who wants to run memory tasks. So that would definitely be a great thing to do, is to sort of have a repository for Mount Sinai's PI's tasks. I think.
If you already have some ideas or questions of what you want to do, or any particular ideas, even if you don't want to mention it here, please email us and we'll open a dialogue and we can get things rolling right now.
Absolutely. And join us on Mondays.
Yeah.
This is very much an open forum.
Any more questions? Anyone thinking of a project and wants to share what you're thinking of? That's a step too far. Okay, great. Thanks so much for coming, everyone. Please visit the website and please get in touch with us and feel free to ask us all kinds of questions. We're really very enthusiastic about this and we'd love to see more people getting involved.
One last thing, sorry. We're gonna keep going with the workshop series, but the format for the future workshop series is not just going to be a discussion. We're actually going to be much more hands-on, almost tutorial style, of how to, I think the next one that we're going to have is basically how to get your data. not only from the XNAT and DICOM format, but how to get your preprocessed data. And we're aware that there's a broad spectrum of experience in Sinai imaging, some advanced users and some people who are just wanting to get started. And our focus is to get as many people in the community as possible. So depending, again, on interest levels and communication that we get, we'll do tutorials. They won't be exhaustive tutorials, but they'll give you the central concepts of what you need to know to do things like anatomical imaging, structural, basically the volumetric imaging. We're going to walk you through everything you need to know, the basics of what you need to know, such that you can take it from there. But we'll tell you the key points, the key tools to use, the key stumbling blocks in using those tools. So that's the kind of structure that we're envisioning for these workshops. So please do keep your eye on the workshop schedule and share this with your colleagues that this resource is coming online.
And if you have something that you're working on that you think other people might be interested in talking about, or you want to get help on, or you want to share, then please let us know that you might like to actually suggest a topic for one of these workshops, because we're always looking for new ideas. Again, we want to make it as relevant as possible to you guys. So whatever you're working on, we want to run a workshop on it, basically. Great. Thanks so much, everyone.